Am I recording? Okay,
Yeah, I think so. I heard the recording progress thing.
sounds good. Thank you. Well,
No
welcome
problem.
everyone tonight. Thanks for coming, and to everyone watching. We're going to go over this week, week 9, where we're going to talk about the regulatory pathways and checkpoints in S phase, G2 phase, and M phase. So just like I believe the past couple of weeks, the general overview slide of the cell cycle. So you have your G1 phase, where you start off the cell cycle with this growth phase or gap phase, and the cell's basically just preparing for DNA replication, making sure that there's enough nutrients, all the cell machinery is intact, no mutations are going on. The cell is also going to commit to the cell cycle in this phase, and then you have the end of the phase checkpoint, that restriction checkpoint, which is where they commit to the cell cycle. It's kind of like the point of no return. Within G1 phase, you could possibly also have G0, where basically it's in a non-dividing state. A lot of our cells in humans are in this state, but that's just something off to the side that's a little bit separate from the cell cycle itself. Then you have your S phase, which stands for synthesis, because you're going to have your DNA replication, and then it also has an end of phase checkpoint. And then the G2 phase, it's growing even more, where it's growing even more, and the cell is just preparing for chromosome segregation. This too also has an end of phase checkpoint before going into the M phase. And then you have the chromosome segregation, of course, in M phase, and the actual cell division. And you have the metaphase checkpoint, or the spindle assembly checkpoint, as it's also known as, within M phase as well. So last week we focused kind of like on the general ideas within each phase, but then this week we're going to really go into the mechanisms of each checkpoint. All right, so starting with S phase, because there's not really we covered a lot of G1 phase last week, so those would be in last week's videos if you need any reviews on that. So starting for S phase, just to remember how we get into S phase, there needs to be a pre-IC assembled, and then the pre-IC comes after the pre-RC. And basically the complex of this comes by due to active CDK. The CDK recruits a bunch of the CDK recruits a bunch of the proteins that are needed to create the pre-IC. And so after the pre-IC forms, you have the polymerase that is recruited in order to start transcription, of course. And so once this polymerase is added, the complex is now known as a replisome. And so once your origin of replication fires, DNA replication begins, you are now surely in S phase, and not all origins fire at the same time. Some can fire a little earlier, some fire a little later, some do fire at the same time. It's kind of specific to the cell. Each cell has a little different method of when they fire and why. And then, so the S phase, you have each phase has specific CDKs. So S phase has SCDKs, as they are known. And so SCDKs are really specific in inhibiting new pre-RCs from forming. And basically, you don't want new pre-RCs from forming because you only want to replicate DNA once. If you have new pre-RCs forming, you're going to have many DNA replication, which obviously could cause a lot of trouble in the cell, because if you're replicating more DNA, you're increasing the chance for DNA damage, which you definitely don't want. So more specifically, what SCDK does is that its mechanism is to phosphorylate the ORC, which is part of the pre-RC, and this is done so by preventing the binding of CDC. And then it can also phosphorylate CDC6, which will cause CDC6 to be degraded. basically when it phosphorylates the CDC6, the ubiquitin is added and then it goes to a proteasome. That's where it's translocated to, so that if there's no CDC6, then MCM can also not be recruited. And then for even more surety that the pre-RC will not form, the S-cyclin CDKs can also phosphorylate MCM27, which is just like the official name for a helicase. And when it phosphorylates this MCM, the MCM is exported through nuclear export. So obviously, if the MCM is in the cytosol and not inside the nucleus, there's no way that it can be recruited to form a pre-RC. And so another big portion of the S phase and what it does is that cohesin rings form during the S phase when the replicated DNAs form. And what the cohesin rings do is that they keep the DNA strands together and throughout the entire duration of M phase. So we'll go into more about the cohesin rings because they play a bigger role during M phase and like metaphase. But it's really important to remember that cohesin rings go on to the sister chromatids during S phase, because there was research that showed that if they are not added during S phase, then it won't work. The process itself just won't work during M phase. And so they again prevent chromosome segregation during spindle assembly and ensures an equal number of chromosome force division. And then the end of S phase checkpoint, it's just to make sure they're checking to see if there's any DNA damage. Is there completely replicated DNA? Is there a need for P53 activation, which can occur? Just to review, P53 is a transcription factor that leads to the transcription of P21, which is a CDK inhibitor. So if CDK is inhibited, it stops the cycle temporarily just to give the cell a chance to fix anything. P53 can also cause programmed cell death. So if there's a damage or a mutation that the cell can't fix, then there is the option of going through apoptosis. And then just in general, staying too long at any checkpoint or stopping at a checkpoint for too long can lead to programmed cell death as well. All right, so before we move on to G2 phase, does anyone have questions about S phase or the mechanisms of S phase? All right, sounds good. So we can just move on. So in G2, the cell is again growing, becoming larger. It's prepping for chromosome segregation now. And it does so by making sure again that DNA is completely done being replicated, that there is complete replication, there's no damage in the DNA, and it will inhibit entry into the M phase until all of the cells until the cell has grown properly to the right size and that all the DNA has been checked. So this of course is really important because if the DNA is not being done replicated and it continues into M phase and continues to divide, then the daughter cells, of course, are not going to have the right amount of DNA or will have damaged DNA and so forth. And so if there's no activation of M phase CDK, which is the result or the product of the end of G2, there is no entry into M phase. So we'll go into now how M phase CDK is activated. So in the beginning of G2 or the G2 checkpoint, MCDK levels are starting to rise, but they are still inactive in G2. And this is because initially you have CAC that will phosphorylate this MCDK with an activating kinase. So CAC gives you the activating kinase, but this kinase is weaker than the inhibiting kinase known as WEE1. And so since it's weaker, even though you have an inhibitory phosphate and an activating phosphate, the inhibitory phosphate kind of wins over the activating phosphate and makes it so that the entire complex is still inactive. And so in order for the In order for M-cyclin CDK to become active, the inhibitory phosphate would have to not be present. It would have to be degraded or taken off. And the reason why, though, that we even want there to be inhibitory phosphate, we want the M-cyclin CDK to be inactive for a little while, is because we need the cell to grow big enough. We need to give G2 kind of like that leeway time to continue to grow and check, just so if you're not large enough, then it would be entering end phase prematurely, which again would cause a lot of problems down the road. So we can't have M-cyclin CDK active just yet, and that's why we have the inhibitory phosphate. So after that checkpoint though, and the cell feels like it's grown large enough that it's done its job checking DNA, that everything's all good, it will continue on into late G2 phase or the end of G2 phase, where it will go on to activate the M-cyclin CDKs. And this is done so by CDC25, which is a activating phosphatase. Basically, CDC25 itself is partially activated at first by another kinase upstream. And you don't have to know anything too specific about that, but just something to keep in back of your mind. And then essentially this partially activated partially activated CDC25 will go and take off that inhibitory phosphate. So that phosphate that was put on by WEE1, it's taken off by CDC25. And so now that there is only a activating phosphate present that was put on by CAC, the M-cyclin CDK is active. What this does though, because CDC25 was still only partially active, MCDK, when it's active, causes a positive feedback loop because you want to have a lot more active M-cyclin CDKs in M phase. So you want to have CDC25 to be fully active instead of partially active. And that's done so through positive feedback. So basically, once your active CDK is active, that the inhibitory phosphate is gone, it's going to go up and positively feedback loop the back into the CDC25. It will phosphorylate the CDC25, and that will cause it to be fully active, which will, of course, cause a lot more rapid activation of more M-cyclin CDK complexes. What an active M-cyclin CDK can also do is it will phosphorylate and inactivate WEE1 just to make sure that WEE1 won't be able to add an inhibitory phosphate again. So basically, it's like inhibiting the inhibitor. But that's the end of G2. Does anyone have questions about that? Let me know if I'm like talking too fast or anything as well, of course. Okay, thank you. All right, we'll keep going on then. So moving on to M phase, just to give a quick overview of M phase as a whole. So you have prophase, prometaphase, metaphase, anaphase, and then telophase / cytokinesis. So in prophase, you have the active MCDK, like we were talking about, that will go in and break down the nuclear envelope, cause the chromosomes to condense, and then start spindle assembly. And in prometaphase, your microtubules / spindles are really growing and shrinking, looking for the right kinetochores to attach to. And of course, we'll go into the different types of microtubules that there are. And then in metaphase, you have the chromosomes starting to line up on that metaphase plate due to a push and pull mechanism that we learned about. And chromosomes will have the proper attachments, make sure that all of the chromosomes are aligned. And then that's what the spindle assembly checkpoint is. It's just making sure that all the spindles are attached, all the chromosomes are where they need to be in order for proper anaphase to occur and proper cell division to occur, in which each daughter cell will have the right amount of DNA that's required. And anaphase, of course, is the separation of duplicated chromosomes. And then in the last telophase in cytokinesis. You have the nuclear envelope reforming, you have your chromosomes decondensing again, and your cell is actually going to divide. And then this is all due to because the level of MCDK. So whereas in prophase you have high MCDK, in the end of M phase, your CDK levels are low again, and that's what's causing all of this to occur and for the cell to be exiting the cell cycle. So we'll go into each one into a little more depth now, starting with prophase. You have active M cyclin CDKs. And what this does is that it breaks down a lot of the nucleus and then just breaks down a lot of the happenings in order for the cell to be open to chromosome formation. So what it'll do is that it'll phosphate the lamin, and the lamin is the nuclear lamina, the outside of the nucleus. It just breaks it down so that there's no barrier between the chromosomes anymore. And it'll also phosphorylate condensin, which will induce chromosome condensation. And I know that Professor Francis likes to say that they go from like spaghetti looking chromatin to sausages where they're much more condensed and like tube like, I suppose. And then it will also phosphorylate various MAPs and motors in order to facilitate mitotic spindle assembly. And we'll talk about MAPs specific motors as well in a couple slides. I think I skipped one. Okay. So one concept that's really important to know as well is sequential phosphorylation. The main gist idea of sequential phosphorylation is that things need to be phosphorylated in a certain order. For example, I believe a couple weeks ago, maybe week 7, we learned about sequential phosphorylation of AKT, which was a certain signaling pathway involved in survival signaling. It had to do with BAD and BCL2, that one. But that was an example of sequential phosphorylation because one thing had to be phosphorylated in order for the next to be phosphorylated. And the same idea here. So in this example, you have the substrate that will eventually bind to PLK1. But before PLK1 can even phosphorylate the substrate, it has to be phosphorylated by CDK. So first CDK phosphorylates the substrate, and then that allows for PLK1 to have a binding site, and then PLK1 will continue to phosphorylate the substrate again. And this is really important to know because remember that CDKs are the master cell cycle regulators. They act as a priming kinase for a lot of downstream kinase. So basically, it primed this substrate in order to be further phosphorylated. And this is important because if you ever needed to stop the cell cycle at any point or at any phase, you can essentially do that by inhibiting a certain type of CDK. Because if you stop a certain type of CDK, then there's no way for further downstream kinases to continue the cycle or to continue the phosphorylation that's needed. So a lot of times in research, you can inhibit CDK in order to prevent further cascades or prevent the cell cycle from continuing at all. So in spindle assembly, we have the microtubules are going to reassemble. So initially in the cell, there already are microtubules, but we basically want to disassemble and break down the entire already existing microtubule network and going to reassemble it into tubulin for the spindle structures. And the spindles are important in order to separate chromosomes eventually during anaphase, but this is the prep for that separation. And so there are 3 different types of microtubules. You have aster microtubules, which are shown in green here. And these basically grow starting from the spindle pole and outwards to attach to a cell cortex. The cell cortex is basically like the inner layer of a cell membrane. And I find that an easy way to remember that would be like an asterisk. So an aster sounds like asterisk, which is like the star symbol on like computers and such. So that's kind of how I remember that. If that'll help any of you guys. Another version or another type of microtubule is the kinetochore. And basically, these grow from the spindle poles and attach to the kinetochores of sister chromatids. So these are the blue ones. So they go from the spindle pole into the middle of the sister chromatids at the centromere where there's a kinetochore. And we'll talk about what kinetochores are in a second as well. And then you have lastly the interpolar microtubules, which grow from the spindle poles and then basically try and match up with other interpolar microtubules from the other spindle pole from opposite ends, and they'll basically overlap. And then these are motors kind of holding them together. And these are all individually really important for creating the structure and making sure that everything is in the cell is properly structured and stable, especially for the chromosomes to line up in the middle during metaphase. Is there any questions so far? All right. So just a brief explanation of what the kinetochore is. You don't have to know a lot about it, but basically it's just a large multi-protein complex built on the centromere of each sister chromatid. So one sister chromatid has one kinetochore, the other also has another kinetochore. It's not just one for both. And it's a really large protein complex, about like 80 different proteins. You definitely don't have to know the specifics of what proteins or how the kinetochores connect, but just that the microtubules will go and try and find the kinetochores and bind to specific proteins within the kinetochore. And then to talk about the motor proteins a little more because of their relation to the microtubules and the spindle. So there is this push-pull model mechanism. And the idea is that spindle formation requires a lot of opposing forces, this pushing and pulling, in order to keep its shape and form. So you have, for example, kinesins, which are positive end motors, and then dyneins, which are negative ended motors. And essentially, they walk towards either the positive end or the minus end of the microtubule. So, if you remember from last week or 2 weeks ago, the positive end is like the polymerizing growing end with the GTP, and then the negative end is more closer to the spindle poles, where like you have the depolymerizing end with the GDP where it's getting hydrolyzed. And basically, this pulling force, these opposing pulling forces, will stabilize the spindles. An example would be egg 5. It's a motor protein that binds to interpolar microtubules. So the interpolar microtubules would be shown here, for example, or this example as well, where you have one microtubule coming from one pole and then another microtubule coming from the opposite pole, and they're meeting and overlapping in the middle. So one motor that could stabilize that and keep them together and hold them together would be egg 5. And egg 5 is a positive end directed motor, which will mean that on one side it is they're both walking towards the positive ends. So that's what's kind of pushing the spindles closer together. And then this is the another overview or like larger picture of this push-pull mechanism model. Right here, we just talked about egg 5, which is like a positive. Sorry, that's not egg 5. These are egg 5s, the positive motors. But an example of a negative directed motor would be dyneins. So, for example, this dynein, they're minus end directed, and one side of it holds onto the cell cortex. So again, like the inner layer of the cell. And then the other end of it walks towards the minus end. So like walks towards the spindle pole. And if you can kind of picture it like tug of war, so it's holding onto this, but it's walking towards the minus end. Oops. It's kind of like it's pulling or tugging in the the microtubule. So that would cause the spindle poles to be pushed further out and like away from each other. So this would be moving this way, and then this spindle pole would be trying to be pushed this way. But at the same time, you have the egg fives, which are pushing it this way and pulling them in. So that's why you have this constant like push and pull mechanism. But essentially, the combination of all of these different motors in different places throughout the entire spindle network, that's what's causing the spindle structure and the proper preparation to eventually pull the chromosomes apart. All right, any questions about spindle assembly or the spindle assembly checkpoint and such like that? All right, thank you. So we'll just move on to metaphase now then. So in metaphase, you have these sister chromatins that are trying to line up. And the way that they line up is through these opposite pulling forces. And remember that as the sister chromatins are all starting to line up and be pushed and pulled this way and that way, they're all secured by the cohesin rings that were added during S phase, just to make sure that none of them get separated from all this pushing and pulling. And so you have the microtubules, that the kinetochore microtubules that want to be attached to the kinetochores of the sister chromatids, and all of the equal and opposing pulling forces are going to eventually push them into the middle of the cell, that metaphase, the supposed like metaphase plate. And because they're equal and opposing forces, that's what causes them to eventually like line up together in the middle. And during this metaphase to anaphase phase, you have the spindle assembly checkpoint to ensure that all sister chromatids are aligned and that the spindles are attached properly at the kinetochore. It's really important because if the spindles are not attached properly, for instance, you could have like one kinetochore with only one microtubule, and then the other kinetochore doesn't have a microtubule. So then that could cause non-disjunction where the sister chromatins do not separate and they all go into one daughter cell. So you would have uneven DNA in each cell, or you could have many microtubules attached to the same kinetochore, and that would basically like rip apart the sister chromatid or the chromosome, and that obviously would not be good for the cell either. So the cell is going to inhibit anaphase unless they know that all the proper microtubule attachments are there. And the way that they know this is that the cell kind of gets like a feeling or a sense. And this is because MAD and BUB, which are just kind of we don't have to go super specific into them, but just know that they're kind of like the main spindle assembly checkpoint proteins. And they will get a sense of kind of like this pulling sensation when they feel like the cell has equal tension pulling from the microtubule on both ends. So if they feel like the microtubules on one side of the metaphase plate is starting to pull in and the microtubules on the other side are starting to pull the other way, they feel like this equal force pulling apart, then they'll know that, okay, all of the chromosomes are lined up. It's now ready to go into anaphase. And that's when MAD and BUB is going to release CDC20. And so CDC20 is released, causing the binding and activation of APC. So, CDC20 will go and bind to APC and then activate APC. And APC is a ubiquitin ligase, which basically means that it's an enzyme that will target further proteins for ubiquitination. And when you ubiquitinate something, you're sending it to the proteasome for degradation. And when this complex is together, what it ubiquinates is specifically securin. And securin right now in normal circumstances is bound to separase. So when securin is bound to separase, it is inhibiting separase's function. So normally you have securin and then you have separase, and normally they're together. And when they're together, that means that separase can't do its job. What separase does is it cleaves the cohesin that has to this point still kept the sister chromatids together. And so when securin or separase are together, separase can't go and cut these bonds. But when you have active APC, this complex, it can go and ubiquitinate securin, cause securin to be degraded, and now separase is by itself all alone. It can go and activate and cleave these cohesin complexes. And then now you have all of these degraded cohesins freeing the different chromosomes for anaphase for the separate chromatids to be able to be pulled apart from each other. So yeah, so once the rings are separated and the sister chromatins can separate, you can now go into anaphase. As a side note as well, APC will also target M-cyclin CDK for degradation. So at the start of anaphase, you start to see that M-cyclin CDK activity, or the level of M-cyclin CDK, is starting to drop. All right, before we go on to anaphase, is there any other questions so far? I feel like that was a lot of information, so any questions? All right, sounds good. So we'll just keep going, flying through. In anaphase, there are two different types of ways that a cell can pull apart the sister chromatins. So you have anaphase A, and then you have anaphase B. In anaphase A, you basically have these chromosomes that are being pulled poleward. So these are your chromosomes, and they're being pulled towards each spindle assembly. Each spindle pole. And this is done so by having the chromosomes be walked towards the poles by negative directed motors. So for instance, diene could be over here on these poles, and they're walking towards the minus end. So as they're walking towards the minus end, and these are walking towards the minus end, you can imagine that these two sister chromatins are going to be pulled apart from each other because of the dienes walking towards the poles, opposite poles. And so your kinetic cores are going to naturally start to shorten, but while all of this is happening, your spindle poles are not changing distance. They're kind of staying in place. And then you could also have, on the other hand, anaphase B, which is when the spindles do grow and they lengthen. The microtubules grow on the plus end. So these are the plus ends, and they're starting to lengthen out. And because these are starting to grow and lengthen, your spindle poles are going to be pushed and pulled apart, even further apart from each other. So that's what this like label 2 is displaying. And essentially, this is also because of a sliding force between the interpolar microtubules from opposite poles. So they're kind of walking in opposite directions, and that's what's causing like the lengthening as well. And yeah, so all of the spindles, the cells are going to grow and lengthen, and then the pulling force is directly on the poles to pull them further apart. It's also important to know that they're not exactly separate from each other, anaphase A and anaphase B. So you can have anaphase A occur first and then anaphase B, or in certain eukaryotes, B occurs and then A, or in some eukaryotes, A and B occur at the same time. So either way, A and B always occur. I don't think there is a situation where only one would occur, but it's just a matter of the type of organism, whether they want to do one first and then the other, or vice versa, or both at the same time. And then last but not least, of course, we have telophase and cytokinesis. So now the sister chromatids have finally reached opposite poles. Yay. You have the cleavage furrow that's going to start forming, obviously, because you want to cut one cell into two. And the cleavage furrow is actually called a contractile ring, or being caused by a contractile ring, which is made up of actin and myosin. which if you know like muscles and stuff like that, that's where a lot of those proteins are. I'm sorry, I don't think they're proteins, but that's where a lot of those actin and myosins are, specific types of cells. And this contractile ring is basically just going to indent the cell membrane in the middle. And then gradually that indentation becomes deeper and deeper until it hits the spindles that are left in the middle, and eventually cut it in half, split it in two, and then they become completely two different cells. And so remember that I said before, when APC was activated, that it was starting to cause the degradation of M-cyclin CDK. So by now, M-cyclin CDK activity level has definitely dropped a lot. And so without a lot of M-cyclin CDK activity, the spindles can start to disassemble. The chromosomes can start to decondense to form chromatin. So you're going from sausages back to like the looser spaghetti like shape. And the nuclear envelope is going to form around the DNA again. And basically, after this occurs, since there is not a lot of M-cyclin CDK anymore, essentially your cycle, the cell cycle as a whole, can start again because you have low CDK activity, which means that your pre-RCs can form again, which means that you can then recruit pre-ICs again, and so on and so forth. Like the cycle just starts again if it needs to. And that is the end of cell cycles. So just a couple of main ideas for like a little bit of a summary. So the cell cycle happens in a strict sequential order. It's not going to jump around or anything. It's very sequential, G1 to S to G2 to M, which obviously M has a bunch of mini phases within itself. And then you have that the cell cycle events involve cyclical activation or deactivation of proteins, which often is through phosphorylation. So, a lot of the main ideas and a lot of the regulation from each checkpoint was dictated by phosphorylation, whether something was actively phosphorylated or inactively phosphorylated, things like that, with like the WEE1 and CAC and such. And then CDK must be bound to cyclin and phosphorylated in order to be fully active. This was kind of like last week's question on the problem set of like, is cyclin necessary or sufficient or both? So cyclin is necessary, but it's not sufficient to activate CDK because it needs to be bound to cyclin and phosphorylated. So cyclin and phosphorylation are both necessary and sufficient, but only one of each would not be sufficient. And then CDK regulates various proteins in different phases of the cell cycle. So different phases of each cell cycle has their own cyclins. You have M cyclin, you have S cyclin, and each do their own things. So for instance, again, S cyclin does all of those phosphorylations to make sure that the pre-RC is not going to form again, to make sure that replication only happens once during one cycle. And lastly, regulation for proper functioning requires activation and deactivation and inhibition. So it requires all of this. Some things need to be activated, some things need to be inhibited. Don't think of inhibition being as negative because sometimes it's not a bad thing to be inhibited, it needs to be inhibited. So it's all about what functioning the cell requires and not that, oh, something that's more is good or something that's less is bad. So that's definitely not the case. But yeah, so I just want to thank you all guys for coming tonight. That's the end. If there are any questions, obviously feel free to ask. But if not, have a good night. And of course, reach out by email to me or any of the other TAs with any questions.
Thank you.
Thank you.
Thank
Thank
you.
you.